Welcome to the first lecture on, or the second lecture on plant diversity, in which we talk about the seed plants. So first off, let's talk about the phylogeny of terrestrial plants. Terrestrial plants appeared in the fossil record sometime between 380 to 360 million years ago. Right, so this was pretty well into the Triassic, but a lot sooner than many people first thought. The terrestrial plant phylogeny is broken up into two distinct clades: the clade of the Gymnosperms and the clade of the Angiosperms. So these are both seed plants. Just a quick review of the plant life cycles, alternation of generations, right? In bryophytes, they were gametophyte dominant, so all the green stuff that you can see is gametophyte, whereas the sporophyte is this little bitty structure, highly reduced, completely dependent on the gametophyte for nutrients. It's in almost every bryophyte, it is non-photosynthetic. So then we had the vascular plants arise. So the vascular plants arose, and these had some changes. One, they became sporophyte dominant. So the largest portion of alternation of generations is occupied, the largest structures are sporophyte structures. So the gametophyte is photosynthetic. It is independent and free living. So they have separate environments from each other. Neither is dependent on the other for nutrients. Then we have the seed plants. So these are vascular plants that have the seed as their dispersal mechanism. Now, these are still, just like the ferns and fern allies, still sporophyte dominant. However, you see that the gametophyte has become highly reduced to the point of being microscopic, and is completely dependent on the sporophyte tissue that surrounds it for nutrition. So obviously not photosynthetic, because it's actually encased inside the sporophyte. Some key adaptations we see in the seed plants. So for one, the sporophyte is highly adapted to the sporophyte is train crash. Some key adaptations. For one, the sporophyte, being the dominant life here, has a gametophyte that's so highly reduced that it is microscopic, as I just said. The spores that are produced by the sporophyte actually stay on the sporophyte. The spores are no longer a method of dispersal. The spores no longer disperse from the sporophyte, right? In the bryophytes and the ferns and fern allies, the spores were released from the sporophyte and were the mechanism of dispersal to spread the plant. Here, since the gametophyte is microscopic and encased by the sporophyte, the spores are not released. They are not the dispersal mechanism. So as I said earlier, it's microscopic. The sporophyte fully encasing the gametophyte allows it to protect that gametophyte against stressors like desiccation, which is drying out, and UV, So it protects the gametophyte from drying out and protects it from UV radiation. And thus the gametophyte becomes completely dependent on that sporophyte for its nutrients. Pretty cool. It's almost the reverse of bryophytes, because in bryophytes the gametophyte was dominant and the sporophyte was reduced and dependent on the gametophyte for nutrients. Well, now the sporophyte is dominant and the gametophyte is highly reduced and dependent on the sporophyte for nutrients. So that's pretty cool. All the seed plants are heterosporous. Hetero meaning different, right? And sporous meaning we're referencing the spores. So essentially, the spores produce the two sexes. So we have megaspores and microspores, right? So anytime you see mega, we're going to essentially be referring to female structures, and micro will be referencing various male structures. So for instance, the megasporangia is the spore-producing structures. Sporangia produces spores. Mega means that we're going to be producing the spores that produce the female reproductive structures. So, megasporangia produces the megasporocyte, sometimes called the megaspore mother. I hate the term megaspore mother cell because megaspore is, its own thing. So I'm going to consistently use the word megasporocyte. And remember, the megasporocyte is diploid. The megasporocyte is what undergoes meiosis in order to produce the actual megaspore. Now, what's fascinating in these seed plants is that meiosis gives you 4 megaspores, but 3 of them become are non-functional. 3 of them are, basically destroyed, recycled, and you get 1 surviving megaspore. As we continue on this path, the megaspore germinates, and we're still inside the sporophyte, man. We're still inside. And the megaspore germinates and gets us a megagametophyte. And the megagametophyte is what, there's my little E. The megagametophyte is what gets us our egg, produced on the archegonium. So, boom. And then we have the microsporangia. So the microsporangia produces the microsporocyte, right? And the microsporocyte undergoes meiosis to get us 4 microspores. Now, all 4 of these survive, right? All 4 microspores survive. And within that, and within this structure, we get the microgametophyte. And inside the microgametophyte, we produce the sperm nucleus. So, the sperm nucleus is contained inside a pollen grain. Wow. I am pressing buttons. It is contained inside the pollen grain. Okay, so we're going to be driving this this little differentiation thing home as much as possible. Now, pollen is a key feature of the seed plants, right? So pollen is our dispersed male reproductive feature. That means that in just about every single seed plant, with, as far as I'm aware, a single exception, all the clades have no flagellated sperm. And we'll mention that exception. So, seed plants do not have flagellated sperm. They do not need water. So, pollen is how we disperse our sperm to where they need to go. We'll mention pollen again shortly. All right, so the megasporangium is worth talking about. So the megasporangium is surrounded by tissue called the integument, right? So this is a thick tissue layer, and it makes up the outside of the ovule. The outer covering of the ovule. So the ovule is our specialized structure which is going to produce the eggs. So the ovule consists of the integument, and the integument surrounds the megasporangium, and inside the megasporangium we find the megasporocyte, which produces the megaspore. And then that megaspore is going to produce the megagametophyte. Oh yeah, I'm driving this whole thing way home. And the megagametophyte produces the egg. So, the female gametophyte, the megagametophyte, develops from the megaspore and produces one or more eggs entirely within the ovule. So, analogous to the ovule is pollen. So, the pollen grain develops from the microspore. So, it's derived from the microspore. Right? The microspore actually produces the pollen wall that contains the structures, right? So inside the microspore wall, we develop a microgametophyte. Oh yeah, this again. And that microgametophyte produces the microgametophyte produces the sperm. Right? So, for every microsporocyte, we get 4 microspores, and all 4 of those microspores have the pollen wall and their own microgametophyte, and they produce sperm. And that sperm is in the form of a sperm nucleus. Right? So we no longer have flagellated sperm, and because we no longer have flagellated sperm, we do not need water for fertilization. Right? So for our pollen dispersal methods, generally it comes in 2 flavors: wind and animal vectors, like our friendly little bee here. Aw, cute little bee covered in plant sperm. Alright, so this is a pretty good video talking about the diversity of pollen grains you can find. The link is right here in the PowerPoints, which, the students on my Blackboard have direct access to. If you happen to have stumbled onto my lecture, welcome, and you can just copy this link right out of the lecture. It's actually a pretty fun talk from a botanist who talks all about all the different kinds of, well, not all the different kinds, but a bunch of different kinds of pollen grains. It's very interesting, but we're not going to inception this and do YouTube inside YouTube, so I'll just skip that video, and we'll move on to seeds. So I already said with the spores developing contained inside the sporophyte, these are no longer our mechanism for dispersal. Seeds are our mechanism for dispersal. Seeds have a large variety of advantages over spores. For one, the seed is better protected, right? The seed wall is derived from the integument. And the big advantage here is that spore walls are generally about a single cell thick, whereas a seed wall derived from sporocyte tissue, the integument, is much thicker, right? So it's thicker, which means it's better able to resist drying out. So it is much better able to resist desiccation than spores. It can last longer than spores before drying out, which is really nice. Seeds also contain a source of nutrients, right? So inside the seed, we have an embryo, which is a multicellular offspring. Right? We are no longer just distributing a single cell. The embryo is actually multicellular. It's generally, you can see the evidence of the roots, and we have the shoots, right? This is the apical meristems, and you'll often have the development of some kind of seed leaf. So that embryonic leaf is called the cotyledon. This is the classic image of a plant, a seedling in its seed for angiosperms. The same concepts apply for gymnosperms, but the leaves don't look the same. So we have that embryo growing, so we don't have to worry about growing from a single cell. And within the seed, we actually have nutrients, right? So this is a nutrient tissue. This nutrient tissue provides both food and humidity to our embryo, giving it essentially water and food so that it can keep itself alive inside the seed. Pretty big advantage over spores. Right? Having your own food and water and protection from desiccation means that seeds are capable of going into dormancy. Dormancy is basically where the plant stops growing, the embryo stops growing. It keeps itself alive off of the nutrients and water contained in the seed and waits, right? It waits for optimum conditions before germination. We can delay germination until we have optimal conditions, which is an extreme advantage over spores. Finally, we have a lot of different methods of dispersal for seeds, right? Spores are always just wind, right? Whereas seeds, we have, of course, wind, if you've ever enjoyed cottonwood season. In addition, we have animal vectors. So that's most likely done through fruit, right? Fruit is a protective covering around the seed, and animals can eat the fruit. And if the seed is adapted for vector distribution, the seed will be protected from the digestive tract and come out in the animal's poop. And what do you know? We've got some fertilizer ready to go. So we've got that humid, nutrient-rich poop and our seeds. It's great, right? We even have mechanical distribution. For instance, those burrs that attach to you when you go for a hike. Those are just so much fun. Even water is a dispersal mechanism. We have our coconut right here. It drops its seed and it rolls off into the water, and the currents disperse it so that coconut trees can colonize multiple different islands. So, clade number 1 that we'll talk about in the seed plants, the Gymnosperms. Gymnosperms are interesting. The classic example of the gymnosperm is the coniferophyte. You probably can guess what a coniferophyte is from the first several letters in there, Conifer, right? So this is Colorado, or I live in Colorado. We've got tons of evergreen trees with our beautiful needles, right? These are gymnosperms. What makes a gymnosperm? Well, one, we completely lack flowers. There is no such thing as a flowering gymnosperm. So, there's no flowers whatsoever. That also means that there's no fruit, right? Fruits are a feature of flowering plants. Now, another interesting thing about our gymnosperms is that they have what we can refer to as an incomplete ovule. Basically, what that means is that our ovule is exposed to the environment, right? And the ovule is not completely surrounded. It's not completely sealed. So if we look at this image here, right, you can see we have the scales. The ovule is located on a scale. So the integument partially surrounds the ovule that is situated on a scale. And thus, this portion of the ovule is exposed to the environment. Okay? If you wanted to take a peek, you can see in this slide, we can reach the ovule. And another feature of gymnosperms not listed here is the strobolus. So a strobolus is essentially a cone-like reproductive structure. All gymnosperms reproduce with a strobolus, right? The pine cone. So they come in two flavors. You have a staminate, which produces pollen, and an ovulate, which contains the ovule and produces the egg and the developing embryo. So, staminate cones and ovulate cones. We use the word strobolus because cones are found in multiple different places, right? It's not all pine cones. For instance, the club moss actually produces its own strobolus, but it's not a gymnosperm in any way. It's a fern ally. This is actually a close-up shot of a pendant I own, and I think it's really cool because we can see a seed or ovule. You know, it's hard to tell with the naked eye. This is basically a pine cone that's been very finely sliced in half, and they polished it and glazed it. So it's really cool. It's easy to see the structure. So this is my ovule sitting on a scale. And if we just look at the scale, we can trace the hollow space, right, all the way out to the external environment of the pine cone. So you can see that we have exposed ovules. Gymnosperm itself means naked seed because it's exposed to the environment. Another picture for a more drastic image of an exposed ovule. And we can compare it to the Angiosperm ovule. So this is the female reproductive structure of a flower. And the ovule is all the way inside, completely encased by an ovary. So it is fully enclosed. There's no exposure to the outside environment in angiosperms. All right, we divide gymnosperms into 4 distinct groups. Coniferophytes, the one you can think of offhand. Cycadophytes, Gnetophytes, and Ginkgophytes. All right, so cycadophytes include the cycads. This is a group of gymnosperms that is not so diverse anymore. They're restricted to tropical environments. Once upon a time, the large forests on Earth were cycad forests. So they look kind of like palm trees. Cycads are cool because they have the largest sperm of every living organism. So it has the most mass. But being tropical plants, they're facing extinction. Hooray! Then we have the Gnetophytes, which are kind of funky looking. The Gnetophytes include Welwitschia and Ephedra. This Welwitschia is nice. You can actually see the strobilus there. Hey, look, strobilus, right? We've got cones on the cycad and the Welwitschia. They look nothing like pine cones. Ephedra is pretty cool. You probably recognize it from, those keep you awake pills truckers used to have. They actually used to call Ephedra Mormon tea. It grows in the deserts of the western U.S. So when the Mormons went to Utah, they found Ephedra plants. And when they brewed a tea from the leaves, wow, it gave them a pick-me-up. So that's kind of cool. And then over here, we have ginkgo, right? Ginkgophyta is probably the least diverse of the gymnosperms because there is exactly one extant species of ginkgophyte. And you probably know the scientific name. If I write ginkgo, you say, yeah, biloba. Ginkgo biloba is the only ginkgophyte. If you look at their leaves, oh, they have 2 distinct lobes. Oh, biloba. Oh, what? Anyway, ginkgos are really interesting because they are the only deciduous gymnosperm. Deciduous simply means that you drop your leaves in the fall. So all the other deciduous plants are angiosperms. Ginkgos drop their leaves. Ginkgos look kind of like they have fruit, but those are not fruit because they are gymnosperms. Only angiosperms have fruit, which is pretty cool. Now, We're going to focus on the Conifer because this is Colorado. And they're the most abundant of all the Gymnosperms. So, conifers are, of course, Heterosporous, which means we have Megasporangium and Microsporangium. Right? And the Megasporangium produces the diploid cell that will become the spores, called the megasporocyte. Megasporocyte undergoes meiosis and produces 4 megaspores. Of those 4 megaspores, 1 survives. Now, I'm not going to say in every single plant 1 megaspore survives and the other 3 break down, but this is the typical life cycle here. So 1 survives and becomes the megagametophyte. The megagametophyte produces the archegonium, and the archegonium produces the egg. What? Okay. Microsporocytes, right? The microsporangium produces the microsporocyte. The microsporocyte undergoes meiosis to get us 4 microspores. And inside each microspore, we develop the microgametophyte, and that microgametophyte produces the sperm nucleus. Whoa. Okay. Woo! Like I said, we're going to repeat this thing. We're getting a little repetitive in the way we repeat ourselves over and over again. Now, there's some interesting adaptations for plants that have a system by which pollen, wind dispersal, is the fertilization mechanism, right? What we have to do is develop defenses against self-pollination. Right? Self-pollination is the plant version of inbreeding. This is not good. Inbreeding can lead to an accumulation of recessive alleles that can produce lethal diseases. Right? So, we have to have mechanisms to defend against inbreeding. In a lot of conifers, the ovulate cones will be higher up in the tree than the staminate cones. Also, if you look at a branch, right, let's get some needles on there. Oh, this is some good drawing here. Right? If you look at a branch, a little deeper onto the branch is where you'll find the ovulate cone. Oh, that's some good drawing. And then it's at the tips of the branches where you find these sort of oblong, paper-like small staminate cones. Right? They have sort of an interesting appearance. Some people have mistaken them for baby female cones, but they are most definitely not. These are cranking out our plant sperm. So if you have wind coming by this branch, it'll grab that pollen when it's ready and blow it out of the pollen. However, you'll rarely have wind that can blow the pollen out and then bring it back in to pollinate the ovulate cone. Another mechanism for protection from inbreeding would be temporal, right? So in one conifer, or not in just one, but in a given tree, you would have the staminate cones mature earlier than the ovulate cones so that the pollen cannot fertilize the ovulate cone. So you can have temporal prevention of inbreeding. Now, one of the fascinating things about reproduction is the pollen tube. So, the pollen tube is how we get the sperm nucleus to the actual egg. Right? So if you look at the ovule with its integument surrounding on the megasporangium, and inside the megasporangium we have that megasporocyte, and that megasporocyte divided and produces the megagametophyte, and the megagametophyte produced the egg. Right, so when that pollen grain is blown in past the scale, it'll land, right? But it's not like there's a mechanism that allows the pollen grain to insert itself in through the tissue of the ovule. So what it does is it grows a pollen tube. So that pollen tube grows into the ovule, and the sperm nucleus gets released to the egg via that pollen tube. So that's pretty cool. So let's talk about our big plant life cycle. That is alternation of generations. So we have 2N and N, our haplodiplant life cycle. Start with our big sporophyte dominant tree. Woo, that's a nice pine tree right there. It's looking good. Okay, how did we get a pine tree? Well, we had a zygote contained inside a seed. And that seed, of course, has its nutrients. So, this is our zygote. And within there, right, our seed fully matured as we developed the actual embryo. I'm not sure what the seed leaf looks like in a conifer, so I'll just make it like this. Why not? Anyway, so this is our embryo. With its nutrient tissue. And once we have a little multicellular embryo in there, this is when we have our mature seed. Alright. And then our seed germinates, and we get our little seedling. Oh, look, it's a little bitty seedling. There's like a branch. There's its little bitty roots. And then it becomes the mighty tree. Alright, so this is all good old-fashioned mitosis happening here. Gotta always remember what's going on. Alright, and then at the end of a branch, so here's our branch. We're going to need to divide this up into, well, maybe I can put it on one. Right? So we'll have our reproductive structure. We have our ovulate cone. And at the tip here we have our staminate cone. Woo! Okay. So, inside our staminate cone, we have a bunch of little microsporocytes. Well, yeah, that's good. Microsporocytes are in the microsporangium. We're not going to label everything. So the microsporocytes undergo meiosis. And meiosis gets us 4 microspores. Oh yeah, we're doing this over and over again. Inside the microspore, and there's our spore wall. We get our microgametophyte, which produces our sperm nucleus. And of course, that, let's see, let's play with colors here. Alright, so this is our microspore. So that's our spore wall. Which will become the pollen wall. Alright. This was our little tissue that was the microgametophyte. Right, so it was mitosis that allowed us to go from microspore to microgametophyte, and it was mitosis that allowed us to go from microgametophyte to sperm nucleus. And this is a pollen grain. Alright, now, inside the ovulate cone, we have our layer of integument. Is not complete. Alright, here is our nice megasporangium. Alright, so technically integument and megasporangium should be like here on the diploid side, but I am totally out of space on the diploid side, man. So, the megasporangium produces the megaspores. We get 4 of them because meiosis, but, you know, only one can survive. So now we'll finally be in our haploid stage. Alright, so there's our integument. Let me put the scale on there. There's our pine cone scale. Very nice. Okay. So, there's our integument. There's our megasporangium. And then our megaspore develops into the mega gametophyte. That looks good. Woo! There. Okay. The megagametophyte produces the egg. Alright. So, meiosis is how we go from megasporocyte to megaspore. From megaspore to megagametophyte, we have mitosis. From megagametophyte to egg, mitosis again. And this is our ovule. So, next thing we need is our pollen grain to arrive. So, much smaller. There's our pollen grain. It's dispersed by wind. It makes it down along the scale, and boop, it arrives. And it produces a specialized structure to reach that egg. And it's this little thing here. And that little thing is called the pollen tube. Oh yeah, I know that there's a picture on the next slide that you can just look at, but if you draw this with me, man, you're going to know it a lot better. Alright. So, we have our sperm nucleus. Inside our pollen grain. Our pollen grain has produced a pollen tube, which meets up with our egg. Right? Fertilization occurs. The sperm nucleus travels down the pollen tube and fuses with the egg nucleus. That fusion is called syngamy. And we now have a zygote. The integument grows and completes over this once we have syngamy, fertilization. And we now have our developing seed with a zygote surrounded by that nutrient tissue. And we get the embryo and woo! Yeah! That is some drawing, people. I did not think I could fit that all on here. So like I said, draw with me, you'll understand it. Although, you might need to draw it a couple of times to fully understand it. Don't just, shoot through this at the speed of me and then expect everything to be fine. Here is the actual pretty picture, right? Our mature sporophyte that's diploid. And on the sporophyte, we have our ovulate cone. And at the tips of a branch, we have the staminate cone. Inside the staminate cone, we have the microsporocytes. And the microsporocytes are inside the microsporangium. So, staminate cone produces the microsporangium. Microsporangium produces the microsporocyte. Microsporocyte undergoes meiosis to get you the microspore, which develops inside the microspore. We get the development of the gametophyte and the sperm nucleus. And thus, the spore wall containing that structure is the pollen grain. Over here in ovulate cone land, we have the megasporangium. Right? So we have the ovule, which is the integument, and megasporangium. And the megasporangium produces, right? The megasporangium produces the megasporocytes. The megasporocyte undergoes meiosis to give us megaspores. The megaspore develops into a very small megagametophyte, which produces a very small archegonium, which gives us an egg nucleus. In the meantime, the pollen grain lands on the exposed part of our incomplete ovule and starts to build a pollen tube, which is pretty much impossible to see in this image. Pollen tube finally reaches the egg nucleus, and fertilization, syngamy, occurs. The sperm nucleus is released through the tube to the egg nucleus, and we get a zygote, which undergoes mitosis to give us an embryo. Once we have that embryo with the food reserves, that nutrient tissue, the integument fuses to create the seed coat, and we have a mature seed, which is then distributed. Now, with gymnosperms, seeds are all wind distributed. This is interesting. We talked about all the cool methods of distribution you can see in seeds over spores, but gymnosperms are purely wind dispersal when it comes to their seeds. So their seeds will often have little structural adaptations to catching the wind, right? You can see the scales have this little seed growing on it, which is pretty cool. It has the wing for helicoptering around in the air. Oh, gymnosperms are down. It's time for angiosperms. Okay, angiosperms. A lot of recent book sources, recent being within the last 10 years, pegged Angiosperm evolution around 140 million years ago. However, in 2013, they found pollen grain in the fossil record containing structural adaptations that indicated that there was an animal vector for the pollen. And again, animal vectors for pollen are angiosperm stuff. So the fact that we found fossil evidence pegging moves back the evolution of flowering plants to something like 265 to 240 million years ago. So this is the oldest known evidence of angiosperm pollen in the fossil record. It's thought that the animal vector was likely a beetle, not a bee. This is because bees are about 100 million years old, and this pollen is around 240 million years old. So, let's see. I think bees are more like 140 million years old. But anyway, that's not exactly tested material right now. Okay, so we are monophyletic with gymnosperms. We have a single common ancestor. there are a variety of shared derived features, synapomorphies, right, that are unique to angiosperms. One, so what divides, right? The shared derived features that differentiate angiosperms from gymnosperms. Okay, one, ovule is completely enclosed. Man, I'm filling up with red ink here. We'll switch to a new color. We have an enclosed ovule. So, we no longer have an incomplete ovule. Right? Another one is that we have two integument layers. Now, two integument layers means we get a much thicker, more protective seed wall. Remember, it's the integument that becomes the seed wall. So two integument layers, which means we get a thicker, more protective seed wall. This allows angiosperms to release their seeds even in crappy conditions. Gymnosperms will usually require specific conditions for an ovulate cone to actually open in order to release seeds, because they only have one integument layer making up the seed wall. For instance, there's a variety of conifers that will only pop their ovulate cones open if you get to temperatures indicative of a forest fire. So, an enclosed ovule, two integument layers to give us a thicker seed wall, right? Oh, a huge one! Angiosperms are flowering plants. So flowers and fruits. This is an angiosperm thing. And another really fascinating one that we will go into detail about is double fertilization. And you know what? If I wanted to, I could even add animal vectors for pollen and seeds for these. Wow, I don't know if that's going to show up. Well, whatever. I know. Lot of writing, but it's okay. Angiosperms are the most diverse of the land plants, right? There's 200, really? I am running out of ink on this slide. Let's see, what happens if we bust this out? There are 250,000 different known species of angiosperms. That's pretty diverse. Gymnosperms? There's actually a little more than 900. Ferns, you have around 11,000. Why are there more ferns and fern allies than gymnosperms? Well, they're tropical. You get a lot of species diversity in tropical regions. So, since they're wholly obligated into tropical regions, you get a fair amount of species diversity. Gymnosperms are often, aside from, as far as the coniferophytes go, the most numerous of all the gymnosperms. You are not occupying your tropical realms, so that's kind of cool. So, 250,000 species, right? That's kind of like 20 times the number of species of gymnosperms and pterophytes combined. Right, so, a lot. And no discussion of angiosperms can be done without talking about the actual flower. So let's talk about the anatomy of the flower. We'll focus at first on the female structures of a flower. So if you look at the female structure of a flower, you have a stalk, a big ovary, and a little site of pollination. This is called the stigma, the site of pollination. The stalk that leads from the stigma down to the ovary is the style. And then the big structure enclosing the ovules is the ovary. This entire structure is referred to as the pistil. Right? Pistil is the more anatomically correct term. Carpel is used in a bunch of sources, but I hate carpel. Dicarpal die. Pistil. Right? So, stigma is the site of pollination. That's where the pollen lands. The ovary is part of the tissue completely enclosing the ovule or ovules. So, the ovary is thickened protective tissue. And it's inside the ovary where we will actually find our ovules. So you'll have a little bit of tissue, and then in a lot of flowering plants you'll have multiple ovules. Right? So the ovules are neat because if you look close at the ovule, one, it's fully enclosed, and two, it's made of 2 layers of integument. Alright, so that is a fancy drawing. Let me try that again. Okay. 1 layer of integument. Oh, my problem is that I am fully enclosing the integument. I'm not going to erase this because warts and all, baby. Okay, so there's my integument. I got my 2 layers. That looks better. And inside those 2 layers of integument are all the standard things, right? We have the megasporangium. Right, and the megasporangium produces the megaspore. And the megaspore undergoes meiosis. Megasporangium produces the megasporocyte, which undergoes meiosis and gets the megaspore. See? Even I get brain twisted on this one, but that's okay. That's okay. Megasporangium produces the megasporocyte, which undergoes meiosis and gets us the megaspore. Only one megaspore survives, and that megaspore produces the megagametophyte. Right, and that megagametophyte produces the archegonium, which produces the egg. Okay, Gymnosperms had an exception to the there's no flagellated sperm, right? Ginkgo biloba had flagellated sperm. Well, guess what? In Angiosperms, it is universal. There are no flagellated sperm whatsoever in the Angiosperms. Flagellated sperm can go swim away. Now, pollen gets to the stigma in a variety of ways. It can be wind dispersed, it can be vector dispersed, but ultimately the pollen lands on the stigma and is deposited mechanically. Mechanical deposition of pollen. So, often by animal, wind too, so pretty cool. And we'll go more into what's going on in here, but let's talk about this ovary because it's a pretty cool new feature. The ovary that completely encloses the ovules. So it's the ovary that completely encloses the ovule. The ovary turns into what we call a fruit. The ovary develops into a fruit. Fruit can mean a lot of things, but ultimately a fruit is specialized tissue that contains seeds. All right? So, here, a tomato is a fruit. Here, a grapefruit is a fruit. Here, a peach is a fruit, and inside we have the seed. The outer covering of a nut is an ovary, right? And then the nut, or the ovary, the nut itself inside a hard shell, right? We have a shell protecting the nut, which is the fruit derived from the ovary. Some ovaries turn into cool, wacky stuff, like the burrs that make your life uncomfortable when you go hiking. Those are great for dispersal. And some fruits look a lot like gymnosperm seeds in that they are adapted for wind dispersal. So fruit is not just tasty berries or, what you think of when you think of fruit. Right? Someone might say intelligence is knowing that tomato is a tomato is a fruit, and wisdom is knowing not to put it in fruit salad. Anyway, fruits are pretty cool. They are often adaptations for dispersal. So, when you bite into that tasty apple, you are squirting the juices of plant ovaries all over your cheeks and mouth. Tasty, tasty plant ovaries. Just biting those ovaries into pieces and crushing those ovaries with your teeth and then just swallowing all that ovary tissue into your stomach. Delicious! Okay. Reproduction, which means we're about to do one of them big old life cycles. So let's talk about the things we need to add to this life cycle. One is double fertilization. So, how does this work? We'll go easy first. Inside the pollen grain of an angiosperm. I just like putting these rectangular things on there for whatever reason. Inside the pollen grain of an angiosperm, we find one sperm nucleus, and then we find a second sperm nucleus. 2 sperm nuclei. So this is going to set us up for double fertilization. Now, let's talk about the ovule of an angiosperm. So we got that double integument layer. Right? Then we have our megasporangium, Oh, yellow is not very visible on this slide. Then we have our megasporangium. Oh, this is looking real good. Nope. Just gonna nope on out that. Try that one more time. Here's our megasporangium. And the megasporangium produces the megasporocytes, which is still part of the diploid phase. Right, here's my megasporocyte. The megasporocyte undergoes meiosis, and we get 4 megaspores. But of those 4 megaspores, only 1 will survive. And that megaspore will germinate, and it will produce the archegonium, and the archegonium will produce the egg, I know, we've done all this before, people. But there is an important note regarding the tissue surrounding the egg. So if we focus in, here's my egg. All the tissue surrounding the egg that will become the nutrient tissue for the developing embryo, right? All of this tissue is what we refer to as binucleate. It's binucleate tissue. This is very important. That pollen grain, it lands on the stigma and it produces its pollen tube all the way down, all the way through the ovary, all the way down to the integument, all the way over to the ovule, right? And that pollen tube connecting up here releases both sperm nuclei. One sperm nuclei fertilizes the egg to get a zygote. The other sperm nuclei fertilizes the binucleate tissue. Those are the 2 fertilization events, resulting in this tissue being effectively triploid. So here's our zygote. We'll make it, you know what, let's make it the full, you know, that contrast is awful. Apparently I am failing school here. Okay, so here's our little embryo, shoot, cotyledons, root, right? And here's my new thick seed coat, because it's derived from 2 integument layers. And then that tissue, right? This tissue surrounding the embryo, which has 3 nuclei in the tissue now. The tissue is trinucleate. This has a name. It is triploid endosperm. And that endosperm is the nutrient source of angiosperms in the seed. So endosperm is the nutrient source. Angiosperms are incredibly diverse. They are the most diverse of all the plants, as we mentioned, because there's over 250,000 species, and you find them on every continent. So you have flowers to attract pollinators, right? That's a pretty huge adaptation. You have fruits to protect the growing embryo, the growing seeds, and those fruits often aid in dispersal. And finally, you have that rich endosperm providing those nutrients. Endosperm is extremely rich in nutrients. So the seeds of an angiosperm are in general much more robust than the seeds of a gymnosperm because they have triploid endosperm, which is high in nutrient content, because they have a thicker seed wall derived from a double integument layer, right? So angiosperm seeds have a lot of advantages, including that, you've got that fruit, that ovary protecting it. So, time for the life cycle. All right. Here's our pretty little angiosperm. Oop, there's a leaf, there's a leaf. Here's a flower. All right, so this is my adult sporophyte. And we got there because we had, here's my seed wall. Here's my zygote. There's my endosperm. Alright, so endosperm. Zygote. Alright. And that developed into the mature seed. That has a full-on embryo in it. Surrounded, of course, by our nutrient-rich endosperm. Which allowed us to germinate when conditions were right and get to our adult sporophyte. So, bloop, bloop. This is my seed right here. It's not a seed until it has an embryo. A zygote is not a full seed. So, there we go. Okay. So let's split this adult sporophyte into our mega and micro stuff. First off, we'll start with our pistil. All right, there we go. So, we have our stigma. We have our style. And we have our ovary. All right, the ovary completely encloses the ovules. That's looking good. So let's get in detail on this ovule. We have 2 integument layers. Alright? Surrounding that integument layers, we have the megasporangium. And so, 2 integument layers. And we have the megasporangium. And the megasporangium produces the megasporocyte. All right. The megasporocyte undergoes meiosis to get us megaspores. So, boop, boop. Bop. And bop. Remember, you get 4 megaspores, but only 1 survives. So we have 1 surviving megaspore. Alright, continuing forward. We get our megaspore developing into the megagametophyte, right? The megagametophyte produces the egg. So here's some megagametophyte. There's our egg. All right. Now, it is important to note in here that we have our binucleate tissue. Egg. Binucleate tissue. So we'll have in that tissue 2 what we call polar nuclei. Oh man. It looks like I am running out of space on this slide. So I'm going to have to draw the male stuff. I think I can get it pretty easy because it's not all that complicated. So let's see how well I can get the male stuff in here. So, over here in the flower, we have a thin little stalk and the male reproductive structure called the anther. Kind of sounds a lot like antheridium, doesn't it? Okay, so inside our anther, we have our microsporangium. And I'm just going to draw a big one, but there's like tons and tons of them, right? We have their tiny, tiny little microscopic structures all through there. But here's my big illustrative one. So, microsporangium. The microsporangium produces the microsporocyte. The microsporocyte undergoes meiosis. And we get a microspore. Well, 4 of them. Each of those 4 microspores, right, becomes a pollen grain. So, each one produces its own little microscopic microgametophyte, which then produces 2 sperm nucleus. So, 2 sperm nuclei. Okay. This is, of course, a pollen grain. The pollen grain lands on the site of pollination, which is the stigma. Remember, this is the site of pollination, not the site of fertilization. Reason for that is because in order to fertilize, we have to grow a big, long pollen tube. So the pollen tube penetrates down into the ovary around to the ovule. So right, my pollen grain goes and lands on the stigma, produces a pollen tube. Right, so here is my pollen tube. And traveling down my pollen tube are my 2 sperm nuclei. One fertilizes the egg. The other combines with the polar nuclei to become the endosperm. So we have double fertilization. Double fertilization, in which sperm nucleus plus egg gets you the zygote, and sperm nucleus plus the binucleate tissue gets us the endosperm. Of course, this can't be a big old alternation of generations without my dividing line. Oh, where am I going to put this sucker? Whee! Oh. Okay. So. It's going to be a tough one. Try this again. Oh yeah, that looks good. 2 in. Oh yeah. I'd just like to take a moment, because we just drew all that stuff. That was insanity, people. There we go. The filled in clades. Right? Chlorophyll alpha and beta. Plasmodesmata, cuticle, right, stomata. Then we have our vascular tissue, sporophyte dominant, seeds, flowers, and fruits. Yay! Done with plant diversity. Ugh.